Okay, so, also guten Abend. Ich werde den Vortrag auf Englisch machen, weil sonst stottert die ganze Zeit. Ich kann nicht mehr zwischen Deutsch und Englisch wechseln, das geht nicht mehr. Aber die Einführung mache ich jetzt auf Deutsch. Mein Name ist Andreas Frutiger, wie Selman schon gesagt hat. Ich doktoriere im Labor für Biosensoren und Bioelectronics und Reatch seit nunmehr 2 Jahren. Ich bin mehr auf der Entwicklerseite, nicht auf der Anwendungsseite, darum liegt mein Hauptschwerpunkt des Wissens mehr in den Technologien selbst als Big Picture. Aber für diesen Vortrag habe ich jetzt trotzdem versucht, dieses Big Picture zu zeichnen. So I'm going to switch to English now. I hope that at the end of the talk you have an understanding of all the words that are listed here. Some of them are quite small, but that you at least have an idea of what each of these terms mean. So as the outline, firstly the motivation, what is actually medical diagnostics, and more specifically what is a biosensor. Then I'm going to move on to give a short, very brief history of medical diagnostics. Then we will see what is easy to detect nowadays and what is still hard. There I would like to show you the success story of DNA sequencing and highlight the challenges that we still have in protein quantification. Then we move on, where is it going? Keyword, point of care medicine. Then we see what do we do with the data, how do we personalize it, what is a healthy human, and how do we make sense of this data. And then we end with the conclusion and we start the interesting part, namely the discussion. If you have any questions during my talk, just interrupt me. I think we can also have like 40 minutes interactive presentation discussion, then go to the real discussion. I think it's more fruitful than just to give a talk first and then discussion. So motivation, what is medical diagnostics? Today, when you have symptoms, you usually go to the doctor and then you ask him, yeah, what is my problem? And he gives you a list. This could it be this and this and this, but he's not sure. In the future it will be different. The problem nowadays is that diagnostics only kicks in when you're already sick in a way. And in the future it should be the way that you don't feel it yourself, but there are tests and data that tells you're sick, you should better visit the doctor or have an MRI scan. This is what we understand under the future of medical diagnostics. So we have measurable quantities, whether this is a protein, a DNA molecule, a glucose molecule, or just a body mass index as a physical parameter. We would like to take these quantities, to collect a huge amount of this, and then from that define what is a healthy human, what is a healthy state. And by machine learning, detect when we are outside of the state, when there are abnormalities. And this is when we then go to the doctor. So that's a big picture. But now we would like to look into how do we measure these quantities actually, and what can we do towards it. So these are categories of markers that we can detect. The first one is DNA and RNA, which is used in order to profile tumors, to identify viruses, that you have a virus infection, and also to classify rare diseases, what kind of mutation we have there. Then we have proteins, for example, for cancer diagnostic, prostate-specific antigen test to detect prostate cancer. You have markers for inflammation, such as C-reactive protein, and for heart attack, you have troponin T as a marker when you can detect whether a person has had a heart attack or not. Then you can detect small molecules like the glucose level in diabetes, lactate, cholesterol, and small ions such as sodium ions. We can also detect cells in tissues directly, leukocytes, so the white blood cell count. We can detect circulating tumor cells, and we also can assess tissue structure and properties via biopsies. And then in addition, this is what nowadays is widespread. We can measure heart rate, body mass index, blood pressure, hemoglobin content, etc. A lot can be done even with watches nowadays, so this watch is constantly monitoring my pulse, and this is really moving into our daily lives. These quantities are measured by something called a biosensor, or biosensors. And I would like to talk a little bit more about these because, as I just said, these are measured, the physiological parameters are measured by classical equipment. So what is a biosensor? A biosensor is something that measures a biological molecule in a body fluid like blood, saliva or urine via a so-called chip surface most of the time. Most of the time it's a chip surface where you have a biological recognition element immobilized to it. And this biological recognition element recognizes any of these quantities and then produces a biological signal that is then transduced via any means, via this surface with a dedicated readout setup that converts it to a signal that we can measure. And this measurable signal could be an optical signal or an electrical signal. So for instance, you could change the local optical properties by adsorption of this molecule, or you can change the resistance or whatever. I don't go into the details now, we can discuss this later on. So how can this look like? How does a biosensor, can a biosensor look like? So at home we're used to these glucose sensors where we have a finger pick and we take the blood, and this is measured by electrochemically by a glucose oxidase which generates electrons, and then we measure the level in the blood. Another very popular at-home diagnostic tests kit are these lateral flow assays. They basically consist of a sample inlet port, then a paper which drags the fluid through reagent-loaded pieces, and then we have a colorimetric readout. We just see by eye whether the color has changed, and then you know whether you're pregnant or not. In the lab it looks a bit different. For instance, you have questions where you would like to know how strong something binds to a protein that you would like to inhibit in pharmaceutical industry, and this is done with a surface plasmon resonance instrument. Or you would like to assess a parameter from the cells in the biology lab, how strong is something expressed, then you usually do this by an ELISA kit. I will talk about ELISA a little bit afterwards. This is really a universal technique. Then in the hospital, if you would like to do genome sequencing, so DNA quantification, you have these genome sequencers, and for protein analysis you have quite huge machines. So this Cobra 8000 is a state-of-the-art immunoassay machine. So a very short history of medical diagnostics. What happened? So everything started basically back in the 1950s. First the DNA structure, when it was resolved. Before, we didn't even know how this looked like, and the protein interactions were only considered in the 1970s as protein interactions and not as only purification artifacts. The first glucose sensor was developed around 1960. In green we have all technological things like sensors. In blue is everything that deals with protein detection, and in red is everything that deals with DNA detection. So we also had the first lateral flow assays already in the 70s, the first pregnancy test, and in terms of protein detection we moved on via radiometric immunoassays, where you measured radioactivity of molecules as a label. Then we had the first enzyme-linked immunoassays, and we started to do analysis of this stuff, and then we moved on to protein microarrays. And the technology I'm working on now is called focal mammography. This is a technology that can overcome a lot of the limitations that classical immunoassays have, as we will see later on, and is considered a breakthrough in protein detection. In DNA, it looks much more promising. We're much more advanced in DNA detection. Polymerase chain reaction was developed in the 1980s, and then we can already sequence whole genomes on top of very small devices I will show you later. So what is easy and what is still hard nowadays in order to detect biomarkers? So easy is to detect anything that is in concentrations in the millimolar or micromolar range. That means something that is present in grams per liter. And what is present in grams per liter? So this can be detected basically by a test strip assay. Just take some urine and dig this into the urine. This is a pH strip. And you can detect the concentration of proteins that are in this concentration range, and also glucose, small molecules, cells, pH, etc., etc. So anything in this concentration range is easy. What has become easy thanks to technology, I already pointed that out, is DNA detection and sequencing. Even though the concentration is extremely low, it's femtomolar to attomolar. So this is 10 to the power of 9 or 12 lower in concentration than the stuff you can detect with these simple test strips. Why can we do that? It's only due to amplification. For the DNA, we have very efficient means in order to amplify the molecules we're after via markers, so-called primers. And this is the technique that was invented in the 80s, this polymerase chain reaction. And it works that you have your DNA piece, you denature it first, you have 2 strands, then you add a primer that binds to these 2 strands, and your DNA polymerase then starts copying it, and you end up with 4 strands. And you repeat this process many, many times. After first cycle, you have 2 strands, second cycle, you have 4, 8, 16, and this is how you can amplify DNA by the polymerase chain reaction. You then, after you have amplified it, have so-called DNA chips. DNA chips, these were invented in the 90s. They have DNA probes immobilized on the surface. These are short sequences of DNA that stand for a disease. So you know that this part of the genome is mutated, so if something binds there, you might have a disease. And you can attach a lot of probes onto one single chip. And they have application in tumor biopsy profiling, single nucleotide polymorphism profiling. That is, a single nucleotide is when like 1 in every 300 bases is altered between all the humans. Some of them don't matter at all, but some of them cause quite troublesome diseases. And you can also have postnatal genetic disease screening in order to identify rare diseases for newborns. And you can even order that. There's a kit for 199 francs on the internet. It's called 23andMe, the company that sells it. You can do this DNA testing by yourself for 200 francs online. But it's not genome sequencing. Genome sequencing is a different story, where the goal is to sequence the entire genome of the organism. And a machine that can do that is, for instance, the Illumina sequencer. This machine was introduced, or types of this machine, the newest one for sure was introduced a few years ago, but the year where this so-called high-throughput sequencing was introduced is around 2006, 2007. And the ones of you that are familiar with Moore's law, this is the cost that it costs in order to sequence a genome over the years. And Moore's law is from electronics. And Moore's law has basically enabled us with technologies like the smartphone or smartwatches or laptops from big transistor circuits. And if you see that this technology also has the same speed as Moore's law, but then suddenly it kicks off when this high throughput sequencing kicks in, and now we're at roughly 1,000 francs that it costs in order to sequence one genome. The principle is, again, you amplify it first, you cut it into small pieces, you sequence these small pieces in parallel, and then you use machine learning algorithms in order to assemble everything again as a one genome. There's a new technology, very recent, which can do sequencing on your mobile phone. This was introduced this summer by a company called Oxford Nanopore Technologies, and this will drop the sequencing costs by another 2 orders of magnitude. So once this is reliable enough, it will cost $10 in order to sequence your genome. And how does it work? The entire technology is based on a nanopore through which the DNA goes through, and on top of this nanopores it's a DNA polymerase. And what you measure is the current that flows through this nanopore. So this nanopore is only a diameter of 100 nanometers, and you have many of these, 100,000 in this chip here. And since each of the bases blocks the current a bit differently, you get a different signal, and you can then identify what kind of base you have. It's not as reliable as high throughput sequencing, but this is definitely the future. Okay, so DNA is easy, but we would like to know the proteins. And protein quantification is a bit tougher. This is the proteasome, the human proteasome in blood. This is the concentration in a logarithmic scale of different proteins that you find in the blood. The classical plasma proteins, albumin, hemoglobin, are in quite high concentration, and the ones which are interesting for diseases are 12 orders of magnitude lower. And the red line is the detection limit of a current enzyme-linked immunosorbent assay. And a few of them are even below that. So for this, you don't need standard assays, you need to heavily optimize that you actually can detect them for larger quantities of blood. The main problem is why protein detection is still hard is you don't have any amplification leads like in DNA detection. You have to actually detect such a low concentration. And this problem can be depicted a bit differently. What does this 1 femtomolar, so 1 picogram per milliliter, mean? It's like finding Nemo in the entire ocean. So if you take the mass of all fish, which is 10 to the power of 12 kilos, and Nemo is 100 grams, it's exactly the same as you would have this huge amount of background proteins and that you would like to find Nemo. So how does current ELISA that reach a detection limit of 3 picogram per milliliter how can I do it? So you start again with the sensor surface, you have your biological recognition element on top of this surface. You add the sample, this could be blood or urine, and then the proteins bind to this chip. The problem that you have, you would like to detect the blue one, but your entire chip surface is covered by anything else that is in the blood. What you then need to do is you need to wash. And after washing, you only have, in the most ideal case, the protein left that you want. And then what you do, you add a second antibody that detects this protein on the other side, but this also binds to the surface where you don't want it to bind, which biases your result, so you need to wash again. And then what you do in the last step, you have an enzyme linked to your antibody which detects the protein. You add a fluorescence, you amplify this signal of the enzyme via fluorophore or chemoluminescence that is created and amplifies your signal. The problem of this entire thing is you need lots of washing, so you will have no chance to implement a an ELISA assay on your mobile phone, because you need a huge tank of liquid, several milliliters, this is a huge tank, in order to run this assay on your mobile phone. So this is not a solution yet, and you also cannot have many tests in parallel, because these reagents might interfere with each other. That is why in hospital you have these huge machines. You need tubes of blood that run in an automated fashion in these machines, and these ELISA immunoassays are executed after each other. As a comparison, your genome sequencer fits on this desktop, and your protein analysis kit that really can do a lot of tests needs an entire room. There was a commercial attempt to actually provide these fingerpick ELISA assays to the public, where you could just have your fingerpick blood and you get all these proteins analyzed in your blood. But within this year, it was a huge fail. We learned that this was a huge fail, and it apparently does not work. The company actually was sued at the beginning of this month for this, and it is really not easy to do all these tests with only a fingerpick of blood, because you might only have approximately 4,000 molecules of the ones that you would like to detect in this drop. That's why you need a larger quantity, and it's also not ideal to use the fingerpick blood, because there you get contamination from the skin and from the body fluid that is in the tissue. You should really get it from the vein itself. But this will be overcome in the future for sure, but it's just a bit too early. Okay, as a summary, what is easy and what is hard? So basically everything except of proteins at the moment is fairly standard, and it is on the way to be miniaturized. So where is it going? I talked about miniaturization, and the keyword for this is point of care. Point of care is enabling diagnosis at the bedside or at home. In order to be able to provide this, we need miniaturized devices. If we look again at the electronics industry, in the 1950s, computers filled entire rooms. And 50s to 60 years later, we have our smartwatches, and the same, I propose, will happen to the immunoassay machines that will end up in a smart lab in approximately 50 years. And how do we see this at the moment? So we already see that DNA technology is already miniaturized to the level of mobile phones. But protein detection is just going to get there. There's a recent paper about HIV and syphilis tests. I will add the reference when I upload it, sorry, I forgot that. And it's really about providing the same functionality as the big machines at home from a fingertip of blood. It's also more going towards the direction of not having discrete data points, but of continuously measuring. You can see this in the case of glucose monitoring. For instance, imagine you're a diabetic, so you take like 5 measurements a day, but the problem is, during these measurements, you don't know what your glucose level is doing, and it might be well off the target zone that you'd like to achieve. So a lot of effort has been spent in order to create a continuous glucose sensor. And Google and Novartis have partnered now to develop a solution that might work. It's a contact lens that continuously measures the glucose concentration in the eye liquid. This is something that could work, because the problem is everything that you put into the body is immediately covered with proteins and cells first, and then afterwards with a fibrinogen cloth. And a lot of sensors fail to work after a few weeks. And this is how it would look like when you have an artificial pancreas. So this division of an artificial pancreas, and a few devices actually came out this year. We'll see how good they work over time. But the idea is that you have a continuous glucose measurement sensor and an insulin pump that is attached to the patient, and you have a feedback loop that you don't have to actively control how much insulin you inject. It will all be automatized. So, to the last topic, the data and personalized medicine. So far I showed you how we can acquire data from measurable quantities, but now is the question, what do we do with it? And how do we deal with it? This is this part. So first, we need to know what is actually healthy, because it's a huge amount of data. It's different kinds of data. It's proteins, it's BMI, it's activity patterns, fluctuations in your heart rate levels, and so on and so forth. And actually there's a lot of effort going on currently to define what's healthy. England is pursuing a 100,000 genomes project, where they try to link genomic sequence data to health records in order to find what mutations cause what kind of illnesses. And also commercial attempts are on the way. Verily, Alphabet company, which is Google, they want to use technology in order to create a true picture of human health. They don't state how exactly they want to do that, but a company founded by Craig Venter, the guy who sequenced the first DNA genome, founded the company Human Longevity, and the mission is that they create the world's largest and most comprehensive database of genome data and clinical data and link it. So basically the same as the Genomics England does. But as you see, proteins so far are still missing. And there are also attempts on the way of that, but this is much more difficult with today's technology than to just sequence the genomes. So how do we make sense of data? And keyword for that is cognitive computing. So we continuously assess the state of individuals and detect abnormalities, but no human will be capable of doing that, because medical data is doubling every 3 or 5 years. People are not so clear which is the case. And it takes approximately 15 years for the latest medical evidence to be put into practice, and also the physicians don't have time to read through all this data that is generated just by researchers. But now imagine I or you as a person are generating continuous data like a jet engine that is supervised all the time. This needs to be completely automatized. And IBM recently in 2011 developed a computer that is a new era of computing. It's like you feed in a lot of data and it builds a model, and it suggests you a hypothesis with evidence. So it says with this and this probability, based on your symptoms, you have this and this disease. And this is actually helping or tried out by oncologists in order to detect cancer. And I would like to show you a video. It's 2 minutes, and based on this, we will then start a discussion that shows how this machine actually works in action, because just explaining doesn't really show how it works. Let's see.
Cancer is the second leading cause of death in the U.S., and in the next decade, demand for treatments is expected to grow by 42%, creating a shortage of 1,500 oncologists. Meanwhile, medical information is doubling every 3 years, making it more difficult to keep up with the latest treatment options. Memorial Sloan Kettering has been training Ibm Watson to help close the gap, as this live system example illustrates. Let's say I'm a clinical oncologist preparing to see a patient. I step into my office, and instead of spending my limited time digging for relevant information, I select my patient from the queue and use Watson to summarize the relevant facts from the electronic health record and identify areas where more information may be helpful. Next, I can match the patient to a clinical trial, or review the suggested treatment plans Watson has presented. Green means higher confidence, while red means lower. To make recommendations, Watson has extracted knowledge from guidelines, best practices, 12 million pages of journal articles, and 200 medical textbooks. Watson provides a visual timeline of the treatment plan, as well as the plan's suggested modalities. Again, they're color-coded by confidence level. Each of the blue icons represents an opportunity to dig deeper into evidence. For example, I can review the specific sources behind each component of Watson's recommendation. Watson has also suggested additional publications for my consideration, an overview of the protocols for administering the treatment, and a summary of information of the suggested drugs. When I'm ready, I can select the treatment information to add to the patient's medical record and share with the patient. This demonstration has shown Watson's ability to create an at-a-glance summary of relevant facts from an electronic health record, to suggest confidence-scored individualized treatment recommendations specific to each patient, and to transparently expose source data that supports the recommendations. Watson is helping oncologists fight cancer.
So this was only for oncology, but this is really how it will be in the future, that you have so much data that humans cannot make sense out of it anymore, and I would say treatment will be guided in a way by machines in order to at least break down the information content such that humans can read it again, or view it again. So, facts and provoking statements, I would say. Medical diagnostics currently is a bit at the stage today as semiconductor industry was in the late 60s. So really at the verge of taking off. Miniaturized chip-based devices, they will enable this point-of-care medicine that everyone is talking about. And the medical data will explode, and we will have devices that will continuously assess our health state and then say when we have to go to the doctor and not our symptoms will tell us anymore. And these routine diagnostics and treatment options, as in the case of Watson, they will become a classification task, which can be tackled by machine learning and automized in that sense. And many nowadays untreatable diseases, they will first become chronic and then eventually become treatable. And many companies are now actually believing to where could this all lead to in another way. Google has founded in 2013 a company that is pursuing the only goal of human lifespan extension based on genomics information and proteomics information of centenarians or just the insight into the aging process itself. Now we really have to ask ourselves, do we want that? And from this we can actually start discussion, I would say. What are your thoughts? Are there any questions of what I've talked about so far? It was a bit hard from the background, what I should do, but let's just start discussing.